Although the heart has its own autorhythmic fibers that allow it to beat on its own and set its own pace, its operation and functioning and how much it beats per minute is still controlled by other factors, one of which is the demand for blood and oxygen by the rest of the body. And in order to meet that demand, the heart has to adjust the amount of blood that it delivers to these different areas. For that reason, we study what we call cardiac output. Cardiac output refers to the amount of blood that the heart ejects or releases into the arteries from the ventricles every minute. This is called cardiac output, or CO. Cardiac output is influenced by two major factors. The first one is stroke volume, or SV. We've seen that term before. This is the amount of blood that is ejected from the ventricle in one beat. So once the heart contracts once, how much blood ends up in the arteries from the ventricles is the stroke volume. And another factor is the heart rate. So how many beats are we talking about per minute? So how much blood is released every time the heart beats? How many times does the heart beat in 1 minute? Multiply those two, you get what we call cardiac output. Now, what goes into this is a lot more information and factors. So we're going to take each of these terms and explore them in a lot more detail. Let's start with stroke volume. What determines how much blood leaves the ventricle during every beat? There are three major factors that influence stroke volume. The first one is preload, the second one is contractility, and the last one is afterload. So starting with the preload refers to the effect of stretching. How much stretching do we have in the muscle fibers, in the ventricles, so in the walls of the ventricles? How much are they stretched? How much force can they generate during each contraction? Usually, the more the heart fills with blood, the higher the pressure and the greater the force of contraction that we get during systole. So let's explore this first factor that we called preload. We know that preload affects stroke volume, and it influences how much blood is released from the ventricle with every beat. And preload refers to the amount of blood that ends up in the ventricles and the amount of stretching that amount of blood exerts on the walls and the muscles. So if there's more of a stretch, more blood, more of a stretch in the cardiac muscle fibers, then the contraction is going to be that much stronger because of the pressure. Think about a rubber band, and you're stretching the rubber band. The more you stretch it, the more forcefully it's going to snap back. Of course, this is all within limits, but basically, the more stretching that you have, so the higher the preload, the more contraction you're going to get, the more blood is forced out of the ventricles with every heartbeat. So that's going to increase stroke volume. Remember, stroke volume refers to how much blood leaves the ventricles with every beat. And so you fill the ventricles up with more blood, there's more stretching, more contraction, more blood will leave the ventricles. The second factor is contractility. Contractility refers to the strength of contraction at any given preload. That means regardless of the amount of blood that is in the ventricles, we also have to take into consideration the strength of contraction. The factors that influence the strength of contraction are known as inotropic agents. Positive inotropic agents increase strength of contraction or contractility. Negative inotropic agents reduce it or decrease the strength of contraction. So let's explore this factor specifically. What are different conditions that can increase the strength of contraction, and what are conditions that will decrease it? So those that increase it, we call them inotropic agents. An example of that would be calcium. So for example, an increase in calcium levels in the interstitial fluid can contribute to an increased strength of contraction because, as you know, calcium is involved in contraction. Another positive inotropic agent would be epinephrine or norepinephrine. Those are going to serve as chemicals that also increase the strength of contraction once we have their secretion, and they would be known as positive inotropic agents, and they are released from the stimulation of the sympathetic division of the autonomic nervous system. Those conditions can increase contractility or strength of contraction, and when you increase the strength of contraction, that means there's more blood that will be sent out to the arteries or expelled from the ventricles. That means we have a higher stroke volume. Some conditions that decrease strength of contraction are increased levels of K+ or potassium, decrease in norepi, decrease in calcium. Maybe there's calcium blockers, acidosis, inhibition of the sympathetic division, as well as some anesthetics. All of these can lead to a decrease in strength of contraction or contractility, which will lead to less blood being ejected from the ventricles and a decrease in stroke volume. The third major component that influences stroke volume is afterload. This factor works in the opposite way compared to the first two. So when you see an increase in afterload, you're going to see a decrease in stroke volume. So what exactly is afterload? Afterload is the pressure that we have to exceed in order for the ejection of the blood from the ventricles to occur. So think about how the ejection of blood from the ventricles to the arteries occurs. In order for the ventricles to release blood into the arteries, the semilunar valves have to open. And in which conditions do these valves open? They only open when the pressure in the ventricle exceeds the pressure in the aorta. And the more blood that you have in the aorta, the higher the pressure. So the ventricle has to even overcome even that pressure. And so the pressure that we have to overcome in order for these valves to open so the ventricle can pump the blood forward is called afterload. And afterload, like I said, has the opposite relationship with stroke volume. So if there's too much pressure in the aorta, that's going to basically increase our afterload. And the more pressure that we have there, we have to overcome it with the pressure from the ventricles. And so the ventricles are not going to be able to send the same amount of blood forward because of it trying to work so hard to overcome that initial pressure. And that would decrease the stroke volume. Which conditions do we see this in? Think of hypertension, elevated blood pressure, or even narrowing of the arteries. All of these contribute to increase in the pressure in these vessels. And when the pressure goes up in the vessel, then the afterload goes up, and in order to overcome that, the ventricles have to work harder, leading to a decrease in stroke volume. So 3 factors again that influence stroke volume are preload, contractility, and afterload. You have several reasons here for each one. What causes it to go up? What causes it to go down? And you know, for preload and contractility, there's a one-to-one relationship. If preload goes up, so does stroke volume. If contractility goes up, so does stroke volume. The only one that's different would be afterload. So in order for stroke volume to go up, afterload has to go down. Cardiac output is influenced by stroke volume, as we've learned so far, and it's also influenced by heart rate. There are many factors that can lead to changes in the heart rate. Changing the heart rate is an important type of control, short-term control, to regulate cardiac output and blood pressure. We know that the SA node initiates the contraction and can set a constant heart rate. It's around 100 beats per minute. But the body needs different amounts of blood depending on the conditions. So the cardiac output and the heart rate has to change depending on those conditions. So let's take a look at the factors that regulate heart rate next. There are several factors here that contribute to a higher or lower heart rate. One of the main ones, of course, is the autonomic nervous system under sympathetic and parasympathetic control. Hormones, changes in ions, age, gender, physical fitness, and temperature are all different factors that can impact heart rate. Let's start with the autonomic nervous system. Nervous system regulation of the heart originates in the cardiovascular center that is found in the medulla oblongata. This region of the brainstem receives information from many sensory receptors, some of which are listed here: proprioceptors, chemoreceptors, and baroreceptors. So think about physical activity, for example, just before it begins. Your anticipation of beginning the activity is going to activate your limbic system, which eventually also sends impulses to your cardiovascular center. As you begin physical activity, the movement of your joints and limbs are monitored through the action of proprioceptors. These are receptors that are found in your joints and limbs and muscles that monitor the position of your arms and legs at all times, for example, just the rest of the body. And the movement of these limbs will activate those receptors that will then activate the cardiovascular center. Chemoreceptors, on the other hand, will monitor the chemical composition of blood. So with increased physical activity, there's more CO2 release into the bloodstream, which makes the blood more acidic. So the acidic conditions in the blood, plus the drop of oxygen, that information will be picked up by chemoreceptors, which will also stimulate the cardiovascular center. And we also have baroreceptors. These will monitor changes in blood pressure, which as you exercise, more and more blood is flowing through the arteries, causing them to stretch more and causing an increase in blood pressure. All of that activates our cardiovascular center, which then stimulates the sympathetic nervous branch to secrete more epinephrine and norepinephrine as well, in order to stimulate a higher force of contraction and then a higher heart rate, which will eventually impact cardiac output. The sympathetic nerves will directly stimulate the increase of the rate of depolarization of the SA node to increase the heart rate, and will influence an increase in contraction of the atria and the ventricles, which again is right here for your information. The parasympathetic response is the opposite. It works on decreasing heart rate by decreasing the rate of depolarization of the SA node, which will lead to a decrease in heart rate and force of contraction. Both these divisions of the autonomic nervous system can work together to regulate heart rate depending on the body's needs. Again, the impulses are sent to the cardiovascular center depending on the changes in the body, and that's where the signals are initiated to either stimulate an increase in heart rate or decrease in heart rate. What you see here with a sympathetic stimulation is faster depolarization of the SA node. You can see here in the graph you have more of those action potentials. They're closer together. With parasympathetic stimulation, we're reducing the depolarizations. There's a little bit of a hyperpolarization condition to slow down the heart rate. We know that the nervous system is not the only thing that can regulate how fast or slow your heart beats, but we also have the action of hormones and ions. Epinephrine and norepinephrine are considered hormones as well that are secreted from the adrenal gland, the medulla of the adrenal gland. We also have thyroid hormones, which an increase in thyroid hormones leads to an increase in pumping effect of the heart, increasing its heart rate and contractility. Cations, on the other hand, like sodium and potassium, have a different effect. If we see elevated levels of potassium or sodium, this can lead to a decrease in heart rate. An increase in calcium compared to the first two ions that I just mentioned right now actually increases the heart rate and strengthens the heartbeat as it's involved in contraction. You might ask, why do these changes or increases in these ions not contribute, all of them, to an increase in heart rate? Well, we've talked about ions and how their specific concentrations around cells is crucial to the production of action potentials in neurons and muscle fibers. So if there's an imbalance, that can quickly compromise things like heart rate. For example, an increase in sodium concentration blocks calcium inflow during action potential of the cardiac muscle. And if you don't have enough calcium, then that decreases the force of contraction. If you have too much K+, then you don't even get an action potential. So an increase in both sodium and potassium leads to a decrease in heart rate because of these effects. However, a moderate increase in calcium can speed up the heart rate and contribute to a higher force of contraction, as we've seen with contractility. We have other factors that can also influence heart rate. Other than the autonomic nervous system, hormones, and ions, we have age. And think about newborn babies. Their resting heart rate is pretty high. It's usually around 120 beats per minute. This declines gradually throughout life. If we think about gender as another factor, females usually have a slightly higher resting heart rate than adult males. Then we can talk about physical fitness. With physical fitness, usually those that exercise pretty regularly have a lower resting heart rate compared to individuals that don't. Fever would be another thing. For example, strenuous exercise or fever can bring up the heart rate because it causes the SA node to fire more quickly, and that can increase the heart rate. And just as heat would increase the heart rate, then if we have a colder temperature, that can slow down the heart rate. We actually use that for our advantage in surgical procedures, especially of surgical repair of heart abnormalities. Usually we slow the patient's heart rate by hypothermia. This is where we on purpose cool down the temperature, and that in turn slows down the activity, the metabolism, the amount of oxygen that the cells need, slowing down the heart, allowing the heart to withstand a short period of reduced blood flow during that procedure. So as you can see, there are many factors like we discussed that can affect heart rate, cause it to increase or decrease. Whatever causes the heart rate to go up, that will cause the cardiac output to go up. Again, one-to-one relationship. And whatever influences the stroke volume to go up will also influence the cardiac output to go up. We discussed many factors that influence stroke volume, contractility, preload and afterload. We can add a few things to that list. And we already learned that stroke volume is equal to end diastolic volume minus end systolic volume. This is how we can calculate that number. And we multiply stroke volume by heart rate to get cardiac output.